So now let's embark on an exploration of all the eukaryotic microorganisms that are typically studied by microbiologists. Keep in mind that we have prokaryotes, eukaryotes, and viruses that we'll be studying throughout this course. So far we've examined the prokaryotes, and now we're going to take a closer look at those that include the fungi, protozoans, and the helminths. So we already have an understanding of how long ago prokaryotic cells evolved, and that was about 3.6 billion years. So it was 1.6 billion years until the first eukaryotic cells on Earth came about. They came about 2 billion years ago. Now, evidence points that these eukaryotic cells evolved from prokaryotic organisms through intercellular symbiosis or endosymbiosis. You probably learned about this in your general biology course. We used to think that the eukaryotes came directly from prokaryotic cells. However, now we understand that they probably evolved from different, more primitive precursor cells that were neither prokaryotic nor eukaryotic. This is what we consider the last common ancestor concept. Under this idea of the last common ancestor and endosymbiosis, some of the eukaryotic organelles that we see inside the eukaryotic cell seem to appear most like primitive cells that are trapped inside. Here's some of the evidence that points to eukaryotic cells arising from this idea of endosymbiosis. You probably know of at least two double membrane bound organelles, the mitochondria and the chloroplasts. Simply, it's suggested that the cells engulfed this more primitive type of cell, and thus this engulfed cell became double membrane bound. We also see that chloroplasts as well as mitochondria have their own DNA, and it's a single circular chromosome, just as we see in many bacteria. It also contains some of the key bacterial sequences. The chloroplasts and mitochondria divide on their own, independently of the eukaryotic cell. As well as that, they contain prokaryotic ribosomes inside the mitochondria and chloroplasts. And as we've already learned, prokaryotic ribosomes are really distinct from eukaryotic ribosomes. So it's suggested that the first eukaryotic cells were probably single-celled and independently living. Eventually, however, they probably formed colonies. And within these colonies, certain cells became specialized for things like feeding, locomotion, or reproduction. After they became this specialized, they lost their ability to exist independently. And thus, we have multicellular organisms arising from these eukaryotic cells. A quick review of how eukaryotic and prokaryotic cells are different. You'll recall that eukaryotic cells are much larger in size. In general, we say about 10 times larger. The prokaryotic cell had no nucleus, no membrane-bound nucleus. However, the eukaryotic cell has a membrane-bound nucleus, and inside that nucleus, we have multiple linear chromosomes of DNA. The various components within the cell allow a division of labor. You'll recall the endomembrane system, where proteins are made and transported out of the cell, for example. There are also other organelles, like our mitochondria and chloroplasts and ribosomes, and cytoskeletal elements that we'll see inside the eukaryotic cell. So in its simplest form, a eukaryotic cell is much more complex and much larger than a prokaryotic cell. This table summarizes the scope of the eukaryotic organisms we'll study in microbiology. We'll have the always unicellular protozoans. Then we'll have some fungi and algae, which could be unicellular or multicellular. And then finally, we'll see some multicellular or helminths. Helminths are considered to be worms, or they have a larval or an egg form, and they infest many animals, so they're of concern for us in this course. Take a look into the form and function of the eukaryotic cell. We'll begin by looking at the external structures, just as we did with prokaryotes. We're going to divide the cell, as you can see in this flow chart here, into external, cell membrane or boundary, and then the internal organelles. You'll notice that there are many more internal organelles to consider in a eukaryotic cell. Now, eukaryotic cells come in a variety of shapes and sizes and are specialized for many different situations, so no one type of cell can really serve as a typical example. So in this case, the cell that you see on your left is a very generalized cell. So it's not specifically any type of cell, it just exhibits all of the features that we might be interested in our study of eukaryotic cells. As we begin at the outside and work our way in, let's look at locomotion. In eukaryotes, we'll see that locomotion occurs either via flagella or cilia. Now, the eukaryotic flagella is very different in structure than the prokaryotic flagella. For one, it's thicker by a factor of 10, and it's structurally much more complex. And this structural complexity is covered by an extension of the cell membrane, so it's actually a component inside of the cell that extends outwards from its perimeter. Now, this flagellum is made of microtubule core. And the microtubule core you can see exhibited down here in this figure. It comes in an arrangement of 9 pairs around the outside and 1 pair in the middle. We see this very same arrangement when we look into cilia. And they're powered by ATP, so it requires energy. And there's these dianin arms that are utilizing the ATP to allow the tubules to slide across each other, which creates flexion and extension, or like a wagging of a tail in these flagella. They're primarily used for locomotion, and it rows more like an oar, whereas the prokaryotic flagellum spins more like a propeller. So again, eukaryotic flagella, very different than prokaryotic flagella. Let's take a closer look now at cilia. They're very similar to flagella, but they're much shorter and more numerous, sometimes in excess of thousands. Cilia beat in coordinated waves, in a rowing action just like flagella, but to move the cell forwards and backwards. We'll only see cilia in a group of protozoans called ciliates, and in certain animal cells, for example, the cells that line our respiratory tract. We'll mostly see cilia functioning as feeding and filtering structures. Most eukaryotic cells also have a glycocalyx. This glycocalyx, or extracellular matrix, is usually composed of polysaccharides. It appears as a network of fibers. Sometimes it can form a slime layer or a capsule. The slime layer forms when these glycocalyx polysaccharide fibers are arranged much more loosely, and it forms a capsule or more solid structure when there's greater organization. This makes phagocytosis much more difficult because there's less flexibility in the membrane. Now, the glycocalyx functions in a number of different manners. First of all, in protection, also in adhesion to other cells and reception of signals that might be coming from other cells. The layer that we see beneath the glycocalyx varies among eukaryotes. Fungi and algae have a thick or rigid cell wall, whereas protozoans and animal cells don't have this sort of cell wall at all. So again, we'll see the cell wall in fungi and algae and in bacteria, but really not in protozoans. It's a rigid structure. It's there to provide support and shape, but it's chemically different from the prokaryotic cell walls. As you can see here, we have the cell membrane of the cell. And outside of that, we have a thick layer of chitin that's linked to the mixed glycans and the glycocalyx by these glycoprotein components. In algae, we have an even different chemical composition. They may contain either cellulose or pectin or a number of other different types of molecules. Now let's take a look at the cytoplasmic membrane. The cytoplasmic membrane is inside the cell wall, and it's a bilayer of phospholipids, very similar in structure to those that we see in prokaryotes. We have there are many protein molecules embedded in the membrane. These proteins allow for functions like transport of materials in and out of the cell through facilitated diffusion. The eukaryotic cell membrane also contains sterols or cholesterols. The cholesterols will add fluidity and stability to the cell membrane, and they're especially important in cells that don't have a cell wall in creating this structure. Eukaryotic cell membranes have many sophisticated mechanisms for transporting nutrients in and wastes out. That means they're selectively permeable, And they'll generally use these cell membrane embedded proteins to help them transport these materials in and out, and they're integral in regulation of homeostasis of that cell. So now we've covered the external structures as well as the boundary of the cell. Let's have a quick concept check. Which group of microbes does not have a cell wall? Is it bacteria, fungi, algae, or protozoans? Well, we just covered that fungi and algae have cell walls. And in the last chapter, we discovered that bacteria do have cell walls, although of varying types. So here the answer has to be protozoans.